Good morning.
Hello.
My name is Ben Novak, and I'm kind of a scientist, so I'm going to be talking to you this morning about the work I do. So, is there any extinct species that you would want to bring back? You?
The dodo.
The dodo is just synonymous with extinction. Dead as a dodo. Here, anybody else? What do you think?
The woolly mammoth.
Woolly mammoth, yeah, there's people working here in Boston on bringing back the woolly mammoth because we have their genome. You?
Dinosaur.
Well, I have bad news. We can't get the DNA from dinosaurs because they died out 66 million years ago. What extinct animal would you bring back?
Passenger.
The passenger pigeon. I'm working to bring back the passenger pigeon. We're gonna be best friends.
This isn't my thing, but I'm gonna do the white rhino.
The northern white rhino? Yeah. So there are only 2 of them left alive on the planet, and they're both females, which means they can't make more babies together.
Do they have to mate?
Well, to make more babies and keep a species going, you have to mate, right? And they can't, there's no boys.
No.
So with biotechnology, we might be able to bring back species. So if we bring back 1 billion birds, how do you feel about that?
We could just pick your lunch up and
What if they pooped on your car?
Ew.
1 billion poops on a car?
That'd be gross. Wow, that's it.
Well, it turns out 1 billion birds pooping in the forest could be a good thing, and that's why we want to bring them back. Can you sound like a passenger pigeon?
This is one of the most genetically diverse rooms in the world. Currently, we have about 11,300 individual cells. They represent over 1,300 species and subspecies of vertebrates.
If you have living cells frozen in a cryotank, then you've already got the whole genome of your organism, and you can use a technology like cloning to bring those cells back to life into a full breathing individual. We're on our way to San Diego Zoo Safari Park, where there's one of the world's first cloned Przewalski's horse, born from one of Revive and Restore's biotech projects for nature conservation.
My father's father came to this land. He rode on the raging tide.
We're gonna get to meet Oliver Ryder. He and Kurt pioneered the idea of the frozen zoo, that we need to save tissues and cells today before species become endangered, before they go extinct.
Hello, Ollie. Oh, hi, boy. Hello, sunflower.
Hello.
Hello, goby. So this is Ollie. He's the second clone of a Przewalski's horse stallion born in the 1970s in the UK. They were one of the first species that was saved by zoos from extinction.
It was really lovely to get to see the kind of bond he has with Oliver Ryder, who he's named for. It's just so fascinating. He was a skin cell in a vial in liquid nitrogen. They take that out and they can make an embryo. And not only is he, this brilliant, vibrant organism, but he's huge. He's a horse.
He's not just beautiful, he's got genetic diversity that will restore genetic diversity that's disappeared from the managed population of Przewalski's horses.
Why does the Przewalski's horse need this type of genetic management?
All Przewalski's horses today descend from 12 individuals that were brought out of the wild. But because we have cells banked, we can provide an assurance that the gene pool of the population can be sustained and can persist.
There are misconceptions about cloning that go back to Dolly the sheep. And we were like, okay, well, if we pick the right species where this could help get more clones in zoos, then people, the public, will be able to see them and know that these are, they're not aberrations of biotechnology. They're beautiful living animals that have a purpose for helping their species.
其实我们也很期望，或者我个人也很有兴趣的一个领域，就是珍稀野生动物的克隆。刚才说到的就是我们的怎么去选择合适的技术路线，我们怎么去选择一个合适的代孕物种，我们怎么去选择一个物种的胚胎。过程中呢，我们一定是沿用一个原则，就是我们让这个数量更多或者是种群更大的动物来去生出来更珍稀或者数量更少的这个动物。比如说我们在前年我们做的这个世界首例克隆北极狼我们用犬来代孕，用犬的卵细胞。现在呢，这个也很有趣，就是北极狼和它的代孕妈妈现在在同一个这个区域里面，然后这个在哈尔滨野生动物园，很有意思，就是在他们的眼中，就是虽然在亲缘关系上，这个代孕的妈妈和这个北极狼是没有亲缘关系的，但是在他们眼中其实还是母女的关系。只要这个物种之前没有被克隆过，没有通过这个技术路线实现，那这个一定还是有很多不确定性的。
I would never do another clone again for myself because it took a toll on me. Part of it was an incredible sadness and a void that needs to be filled. And part of it was not understanding what a clone is. Khan was born in China in the laboratory. Singapore law does not allow dogs to be in Singapore unless they had 6 months in China after the rabies vaccination. So by the time I got him, he was nearly a year old, he was 11 months old. In the beginning, for the first 4 to 5 months, it was difficult to recognize Khan. The anxiety part is one that I was most concerned about. The behavior was totally abnormal. He would nip and bite for no reason, and he was full of fear and anxiety. It was very hard on many levels because I got so many people who are against it. And I had letters written to me from my association telling me that what I'm doing is wrong, is unethical. So of course, I start questioning myself and my motives. I was very conflicted. Have I done the right thing?
All identical twins are clones. They're not clones that we artificially made, but they are clones. And if you've ever known identical twins, you know that they're not the same person because of things that happened to them either before they were born in utero or happened to them afterwards.
Khan really needed rehabilitation. And the best rehabilitation for Khan was socialization. So I left him with this wonderful woman who helps to rehabilitate dogs. And it is where he learned respect, he learned boundaries, he learned about who he is, and who he was gone. It's only recently Khan actually appeared through him. Yes, I'm here. Every day is a new experience, a discovery of the old Khan again. Every day, every week, every month, I'm getting bits of Khan back. I've started recognizing Khan more and more in this clone Khan. I think I have my Khan back.
The main idea behind the ecosystems restoration that it's super complicated and super hard in the initial stage. Where we want to get is that we want our ecosystem to become self-sustainable for humans, for animals, for nature, for climate. Right now, we are inside the sediments, which are in Russian called вечная мерзлота, which is meant eternally frozen. So most of the ground in the Arctic are underlain by permafrost.
You don't really understand what permafrost is until you go underground. Well, traditionally in that part of Siberia, if you go fishing in the summer, you dig a tunnel into the ground and you take your fish down there. It's a freezer, it's a free freezer, no electricity needed. And so Zimov started building one in the park. I think he had several motivations. One, they store food there, like stockpiles of it, for in case they need it in the future. And then he also wanted to do research down there. He wanted to be able to pick the soils from the various depths and analyze it to see, like, what was the ecosystem here, what was the carbon content, what plants were here.
So you see these tiny, not even tiny, like roots of grasses, and that's the grass which mammoths ate. And the age of these roots, I think around here it's about 30,000 years. So that's roots which preserved for many, many millennia. So overall, the storage of carbon in permafrost is huge. There is more than 1,000 billions of tons of carbon stored in permafrost throughout the world. That's way more than stored in all above ground vegetation of the planet. The problem that as climate is getting warmer, it's maybe that these frozen grounds will not be frozen for eternity anymore. And the issue that if this permafrost will store, all these roots will be quickly decomposed by microbes and converted into greenhouse gases.
Permafrost is a huge climate feedback loop. The more permafrost melts, the more greenhouse gases it releases, which warm the climate, warm the atmosphere, which causes more melting and more greenhouse gases. It spirals out of control. In the town of Przewalski, the town where they live, there's buildings where there's just like a 20-meter deep hole opened up under the building. Like, things just collapse.
Well, that used to be a, what's called, a refinery. And then I think over the course of 1 year it collapsed. And I think in the next 30 years, pretty much our entire town is, and that region, those big buildings will also start looking approximately the same. the question is, how can we prevent that from happening? How can we keep permafrost intact?
There's actually like several mechanisms, but the biggest one, the most important one, is snow removal. So the snow, it's like a down comforter, like a big, warm, fluffy comforter spread over the entire Arctic, and it's keeping the ground warm. And if I'm a bison and I'm hungry, I want to eat something, and I start digging through the snow, bison do it with their heads like this. They like push the snow away. By moving the snow to get the grass underneath, you're bringing this cold air in contact with the ground and cooling the ground off.
The Zimovs have shown that when the animals are present and the snow is scraped away, they do get much colder sediments, and they're hopeful that this could actually help trap some of that methane that they know is being released constantly into the atmosphere because of melting permafrost.
So why hasn't Pleistocene Park been replicated? It's fairly labor intensive to do it. Also, I think it hasn't been totally proven, right? So it's a hypothesis that has very good theoretical foundations, but until that's really firm, in a way, there's nothing to replicate.
Realistically, the northern white rhino is extinct because they can't rescue themselves anymore. There are only 2 infertile females left. And then we published that we are capable to produce viable embryos, which have a very high potential to produce offspring if you transfer them into a surrogate.
Geht's denn jetzt weiter in Kenia?
Embryotransfer selber, der oft ein Problem ist, der war in 20 Minuten vorbei. Wir haben ja nur einen Embryotransfer gemacht.
Das heißt, du musst dann nochmal hin?
Richtig. Aber ob dieser Embryotransfer jetzt stattfindet, hängt ein bisschen von der Interaktion zwischen den Bullen und der Leihmutter ab. Jetzt müssen wir warten.
Dann hoffen wir, dass es gut geht.
Ja. We had clear evidence that the embryo transfer is working with southern white rhino embryos. However, we have no idea what a northern white rhino embryo is doing in a southern white rhino uterus, because they are nearly different species.
This entire project is a race against time, especially because we're working with a subspecies that only has 2 living individuals, which are not very young. So we came in very late in this race, and but we will still hope to win it.
The next thing, if everything went well, we might see a northern white rhino, I don't know, hopefully in the early 2026 to be born. But I would still regard it as a miracle.
To have a calf on the ground, it's only the first step of the entire program, because we still have a very narrow gene pool.
We have 1 female, and so far we did embryos from a sperm of 2 males. So founding population at this moment would be just 3 individuals.
So here you see skin cells of the northern white rhino fatu. So I started a reprogramming experiment where I want to reprogram skin cells to stem cells, and this is now 2 weeks after I added the reprogramming factors, and now I see that there are colony formed. So this is like the typical stem cell morphology.
From this stem cell, in theory, it's possible to produce gamete, so it means egg or sperm in the lab. And if you have egg and sperm, then you make an embryo, and then the rest is the same.
I think this is also fundamental important discovery, not only that you can derive oocytes from female cells, but you can also do it from male cells.
And all of these technologies will revolutionize how we do biodiversity conservation today. Imagine being able to make more white rhinos without needing a white rhino.
Это одна из крупных, когда-либо обнаруженных целых ног мамонтов. В прошлом году он был обнаружен. Основные кости они находились на поверхности. Две конечности они были в мерзлоте, поэтому сохранность такая. Останки мамонта были обнаружены в прошлом году на острове Большой Ляховский местными жителями, которые занимаются сбором бивня мамонта. Можно даже утвердить, что это взрослый самец, потому что только взрослым самцам могут принадлежать такие крупные конечности. Вот ногтевые пластинки. Сколько? Вот тоже, да? Она. Раз, два, три, четыре. Вот она идет. Видишь, первая. Ну, проект по возрождению мамонта, он сам по себе такой один из амбициозных проектов. Вот, я отношусь к тому числу научных сотрудников, которые надеются увидеть живого мамонта.
И как раз наши партнеры из Японии и из Южной Кореи как раз бросились клонировать мамонта. А давайте вот попробуем вытащить ядро из клетки и вот пересядем, ну вот и пересадим его в клетку, ну вот если клетку слонихи вот индийской, и получим мамонта. Мы хотим вот найти действительно хорошо сохранившегося мамонта, получить, ну вот наконец возможность все-таки попробовать склонировать, а вдруг получится.
It's true that some single-celled organisms might freeze in such a way that they can be resurrected and still be alive. And it's true that in a lab, you can actually take small animals and freeze them in such a way that they can be resurrected. But the possibility that a single cell in a mammoth at some point froze in exactly the right way, that it can just be brought back to life, I mean, never say never to anything, but yeah, really, really unlikely.
Ну, мы сейчас возьмем на ДНК-анализ небольшой кусочек.
Когда стало понятно, что с клонированием появились трудности, ну вот появилась новая возможность. И мы вот начали вот работать уже с американцами. Они у нас взяли вот образцы и начали оттуда вот вытаскивать именно специфические гены мамонта.
Это редактирование генома. И редактирование генома на сегодняшний момент считается наиболее вероятным вот методом, при котором мы увидим не живого мамонта, а редактированного слона, адаптированного выжить в наших суровых условиях.
So, how do we bring back a species like the mammoth? Our stated goal is to bring back a cold-resistant elephant initially. This still involves an unprecedented amount of genome engineering, but it could be as few as 100 gene edits, which is comparable to the number we made in pigs, or it could be more. We'll find out.
We're taking an elephant and we're making a little bit more mammoth-like. We would need to understand, first of all, what it is that makes a mammoth a mammoth. The hairier, external surfaces, the slightly longer, more curved tusks, the different body shape. What are the DNA sequences that actually create those phenotypes and behaviors that are missing? After we identify those, we would take an elephant egg cell, we would remove the genetic material, the nucleus, from that elephant egg cell, and put in its place the nucleus, the genetic material, from the cell that we had been editing in the lab. So that, when it was then transformed into an embryo and implanted in a surrogate host, would eventually develop into our mammoth, or our Arctic adapted elephant, if you prefer.
So my background is in building technology companies with much smarter women and men than me. And so I said, George, if you had one project that you thought you had unlimited capital for and you could just go pursue at all costs, what would it be? And he said, I would bring back the woolly mammoth, revitalize the ecosystem of the tundra, and build technologies that could help conservation. And in that moment, it was like a light bulb went off and I was hooked.
So what's in this? We were essentially on a starvation life support until Ben arrived in 2021. He said, you know, this is really a great project, you should have funding for it. I said, oh, that would be nice. And he said, well, I'll help you raise $2 million. I say, oh, that's amazing. I don't know how you're gonna do that. I had felt that it was unfundable, but I was completely wrong, and Ben's intuition was much better.
So Colossal chose 3 species, the mammoth, the dodo, and the thylacine, as the target species for de-extinction. What's good about these is that we have 1 placental mammal, 1 marsupial mammal, and 1 bird. And so as we develop technologies along the path to these 3, we are developing technologies that are applicable to quite a large range of the animal tree of life that can be used for other projects.
When you're doing genetic engineering, you're doing it on a cell in the lab. But then you have to take that cell and you have to turn it into an animal, right? But for big species, like mammoths or elephants, that creates new problems. Elephants are endangered. You don't want to use an elephant as a surrogate mother, right? It's too big. The public wouldn't like this idea.
We are in the early stages of the artificial womb. I do not believe our first generation animals will be born ex utero, but I do think eventually they will.
With marsupials, the artificial womb technology only has to keep this animal alive for 12.5 days. And so that seems much more doable than 22 months of gestation with a placenta for a mammoth or an elephant.
So the question is, are we still on track for delivering a genetically engineered elephant by 2028? The answer is that I think we still are on track for 2028.
Is that realistic? My instinct tells me no. I don't think this is going to work. But now they have millions of dollars to prove people like me wrong.
Imagine that de-extinction worked, and you get a baby woolly mammoth, born from an Asian elephant mother. How does she teach it how to be a mammoth? She doesn't know how to be a mammoth.
The first few will simply be edited elephants, and they will stay with whatever herd their mother is in. And they will presumably be recognized as her child. Then eventually, once they are proven cold resistant, then they will be put in carbon-rich parts of and low human population density parts of the Arctic.
Will the first mammoths be released here in the park or in some other place? The problem that ecological effect from mammoths will not happen in the next probably 50 years, because you need more than one mammoth, you need the population. And so that's a kind of many decades / centuries problem, which, you know, the fact that it's a century-long problem doesn't mean that you don't have to do it.
The technology which leads to the mammoths is a very essential and very vital technology which we can use to make our northern white rhinos better, because there might be about 150 northern white rhinos sitting in different museums in the world, representing the genetic code of animals which lived 100 or 200 years ago.
Ja, klar. Ja, die kommen sie gerade.
Achso, da. Hier am Bauchfutter hängst du. We didn't destroy the skull from outside, so the museum is still happy. If we can encrypt this code and use the technology developed by colossal scientists, then we can utilize this dead animal to make gametes. That would really widen our genetic diversity in an unexpected way.
One, we're actually the genetic rescue partner for the northern white rhinos. And so we think that there needs to be a bigger initiative around what we call biovaults versus biobanking, right? Taking frozen tissue cells and just labeling it and putting it in a freezer, that isn't really biobanking the way we define it. We need to take lots of tissue samples. We need to build population genomics maps. And then we also need to establish immortalized cell lines. And we need to build tissue banks. And we also need to create induced pluripotent stem cells. So there needs to be this concerted effort into something that's much broader. And that requires technology and funding.
If you're worried about species becoming extinct, you have to worry that a senator from Oklahoma or the government of Tamil Nadu will say, you know, we could really use this new factory. It'll provide a lot of jobs. It will help our economy enormously. Yes, it may drive this little frog extinct, but even if it drives the frog extinct, we'll just save some DNA and we can bring it back later. That's the moral hazard argument. And it's not a crazy one, and it's really hard to evaluate.
It's so great to see you.
It's great to see you.
It's been a really long time.
Well, when we met, I think you had this vision for like where the work was going to go and where it would take you. So since then, have things unfolded in the way that you thought, or has there been real changes to, you know?
As an organization, we grew from the passenger pigeon and to now funding nearly 80 projects around the world. But for the passenger pigeon project, that did not translate to dollars at all. And that all changed 2 years ago. So we do have now a anonymous donor that's putting enough in to get us all the way to the birds. So technically, the vision I had when we met 10 years ago, it hasn't really changed, but it's finally
you're getting the traction.
Finally going. It's finally taking flight. And
in the case of the passenger pigeon, we started with a small amount of funding, you know, $25,000 grant on this, $50,000 on that. So many people asked me when I was starting Revive and Restore, why was I choosing to do it as a nonprofit? And I was pretty adamant that it had to be a nonprofit. In a controversial field like genomic engineering, we needed to be beyond reproach. That any IP that would be created along the way would be open and available for the use for wildlife conservation.
how do we raise the first generation of new passenger pigeons? So we're gonna gene edit them, give them the genes they need to be hypersocial. This is what I want to do with you, possibly here in the forest you've got. Because to raise the first generation of passenger pigeons, we need surrogate parents.
Right.
And so
I can certainly donate a portion of this 3 acres in the woods, so they have the environment of the wilderness, and then introduce the columbolivia, a standard rock pigeon, use them as surrogate parents.
But they grew up originally in flocks of hundreds of millions to a billion, right?
Right.
That sound, that noise. So we need speaker systems rigged up, playing the sounds of a billion birds. And we want maybe a mural in the background wherein they see the images of thousands and thousands of birds. We can use decoys, but basically we want these new chicks to be growing up in an environment where they're seeing and hearing millions of birds. And the reason this will work, it sounds kind of crazy to raise a bunch of birds with a bunch of fake decoys and speakers, but this is actually how the restoration of seabird colonies works. I want to be able to get to a point where we produce more than 10,000 chicks a year, which means I need 20,000 parents minimum, 10,000 moms, 10,000 dads. And I need an aviary to house minimum 20 to 40,000 passenger pigeons. But if we build it in a way that people can actually come and interact, then people will see the promise of why we want the birds back. But if we don't do it...
The most fascinating thing for me about the passenger pigeon is this idea that it might have played a very particular function on the landscape as a sort of force of disturbance. So does our society today have the capacity to tolerate these forces that are ecologically healthy, but socially we don't have the ability to really make space for them?
What human beings can do in the span of a year, the pigeons did in 3 days. And they went to a new spot for a few weeks and did it there, and then a new spot and did it there. The pigeons were the real source of this incredible ecosystem engineering service. And it'll take more than a century to probably get up to millions and millions of birds. So I do think there will be a lot of time to learn how to deal with that recovery and to coexist.
Well, I'll see it myself. Highly unlikely. I'm 73 years old. But anyway, my grandchildren, my great-grandchildren, hopefully they'll see the bird incarnate. And I'm going to do everything I can to have that happen.
My kind of dream in the future is that this is all going to work. And we're going to come to a place like this where the autumn colors are changing. But I'll be able to look out and the sun will be setting and I'll hear the rustle. And I'll look up in the sky, and in the changing, fading light, I'll see them. There will be our birds.
These instruments are super rare. There is, I think, overall in Russia, I think now there is less than 10 sets of such instruments operating with full methane systems. So there is very sparse information on what's happening out there in the Arctic, how permafrost is changing. And it's super critical for us to maintain those instruments running.
Things like this, like permafrost, how much is it warming up? Or plants are growing faster, which is absorbing CO2, but then decomposition is speeding up, which is releasing CO2. How do these things balance out?
So that's CO2 concentration, methane concentration, so there's main two greenhouse gases. And knowing where the wind is going and how the concentrations of greenhouse gas change, we can see if, let's say, the ecosystem where we are now either kind of breathing out greenhouse gas or absorbing. Well, it's, you know, we are doing fundamental science in the high Arctic in Russia. Like, it's the least profitable place and the least profitable business. Let's say last years are tough. First, the COVID was not helping, and then the situation in Ukraine and all the sanctions are really restricting. So here in this file we have all the data which we ran this spring and also from 2018. Well, the good thing that within the park we have a very good results, and we actually still sequestering carbon there, and the rates what we see is quite high. So the effect of animals is positive. But with temperature of permafrost, that's actually the most complex effect to have, because you need many animals, and also you must have predators. Because if you won't have predators, your animals will not evenly move, and you will have overgrazing in this place, some place will untouched. And that's kind of one of the kind of future goal of development of the park, like next stage, is to actually get enough territory, enough herbivores to be able to introduce some predators. That's kind of the must-have.
They're really, you know, very scientific in their approach, but they have to be like cowboys to get the science done. They have to herd bison and caribou, elk, and shaggy horses, and so on. And so they really are some of the last real, truly free cowboys. But they can still be rigorous scientists at the same time. So they're my heroes. I mean, they really are fighting the good fight.
When I met Nikita, he wasn't a rescuer of the world. He was just a student who played basketball. He told us all those stories about Pleistocene Park, but it was like stories about other planets. It's not about believing. This is life, what we live. We earn money and we spend it on Pleistocene Park. We have time, we spend it on Pleistocene Park. It is life story, it's not a project. Так, наверное, надо открыть.
Сейчас крышки сдует. Настя, Сашка, лови. Ну, давай, ладно, молчи.
Now, ironically, I'm trying to replicate Pleistocene Park in Alaska. Yeah, so it's kind of literally become my story, and we'll see where that goes.
Of course, I doubt that I will be able to create the world's largest ecosystem by the end of my lifetime, but at least it would be nice to get it on its trail, where it can get there itself, naturally.
So this story is like a movie, and what I envision as it has no end. 20 years from now, you're not talking about extinction or saving a northern white rhino. You're using that technique to save other species that are threatened or that are near extinction.
What would it take to not just save a species so that we can go marvel at it from the other side of a plate of glass, but actually save the species so that it can thrive on this earth?
I think anyone working on de-extinction projects these days needs to keep a focus on what they're trying to bring back. Is it going to be fair to that animal to bring it back into the world that we're in now?
Sometimes people get caught up in the semantics of de-extinction versus species preservation versus cloning versus resurrection, but at the end of the day, we are taking dead and extinct genes and lost biodiversity, which is so critical to what we need for the future, and bringing that back.
If the question is de-extinction hubris or hope? I think the only honest answer is yes. It's a little bit of both.
And that's why I'm interested in de-extinction of species, because all these technologies that you need, stem cells, artificial eggs, artificial wombs, but if those things could work with a mammoth, then you could also produce humans that way, right? That's why it's interesting to me, not because of the woolly mammoth.
The direwolves is the first species that we're bringing back at Colossal.
When we edit different lines of genetically diverse gray wolves, we'll create a set of genetically diverse direwolves.
And we take our gene editing tools and we go into that cell and we cut out the bit of DNA that looks like a gray wolf and paste in its place the DNA that looks like a direwolf.
That cell becomes an embryo that's a direwolf.
To bring something back from extinction, it's a transformational moment in human history, right?